Now, okay, so let's set up our cell, and I'm going to draw this really, really big because, of course, having plenty of room to draw is going to be important here. So yeah, that's quite large. And I'm going to draw the adjacent cells too, but we're really not going to do anything with them. It's just they're there for context so that you know that the cell I'm going to focus on is not the only one here. And actually, I'm going to erase something really quick because I would like to have more room. So I'm going to make this cell a little stubbier just so I have space to draw. There we go. That ought to do it. And of course, I'm going to label stuff, so don't panic. We'll get those soon. Okay, so let's do some labeling. I'm going to label in the upper left-hand corner. So all of these are, this is the proximal, convoluted tubule. So we see the suffix ule, which means itty bitty tiny little tube. So of course, these are simple cuboidal epithelial tubes that are all wadded up, and that's mostly what the kidney is made of. But since they are just one cell layer thick, that means that they're only observable microscopically, hence tubule, right? And so this is one simple cuboidal epithelial PCT cell. And I don't have really any applications open, so I'm not sure why I'm getting a low system resources warning, but hopefully it'll work out. Okay. And then down here, I'm going to label my peritubular capillary. So as always, paying attention to the actual meaning of words and not just be like, well, that series of sounds means this is important. So peri means around. So peritubular means around tubes. And then we know what a capillary is, right? So the name for this thing is telling you this capillary network is wrapped around parts of the renal tubule, specifically because tubular reabsorption and secretion means exchange of tubular fluid components with the blood. So sometimes you want to take stuff back from the tubular fluid and put it in the blood. And sometimes you want to put stuff from the blood into the tubular fluid. So that's what reabsorption and secretion refer to. But, you know, we require proximity of the blood in order for any of that to work, right? So we've got to have capillaries nearby. So as I was saying before I started this, we have a familiar pump to think about in a familiar role, and I'm going to make it kind of big just to emphasize its importance and also the fact that you know what this is. It's not a stranger to you. So it's an ATPase, meaning it spends ATP to do some work. In this case, that work is active transport. So it's going to be moving two things in opposite directions. So every time it spends an ATP, it's going to move to potassium in, and we're going to move 3 sodium out. So the reality is at the basal surface of each of the PCT cells in the tubule, we're going to have lots and lots and lots of this pump, but I'm only going to draw one of them because I need to have room to draw the transport of other things. So just mental note, expect that the basal surface of all of these cells has many of these pumps per cell because they're doing an important thing. So this is active transport, obviously, because we're spending an ATP. So really what that looks like is something along the lines of this, where we have ATP and then boop, it becomes ADP plus inorganic phosphate. And then in cleaving that phosphate off, we release a little blurb of energy that's used to do work. In this case, pump things. So we know that this pump is really good at setting up concentration gradients, and we know that because we learned it in the context of neurons and muscle cells and other things, but now it's doing the same thing in a kidney cell. So what this is doing, if you imagine that you have oodles and oodles of these pumps at the basal surface, not at the top, but only at the bottom, that means that we're constantly pumping sodium out of the PCT cell. So if we're always, always, always pumping it out, that's going to create a situation where the concentration of sodium inside of the PCT cell is low due to this pump. Because as soon as we get any in, we're just going to move it right back out. So we have a concentration gradient set up, and it's a pretty steep one because we're really aggressively moving sodium out of this cell, so that there's always going to be really low sodium in this cell and really high sodium in other places, which is great because we know that the steeper a concentration gradient is, the faster the diffusion of that thing will occur, right? So that means two things. One, if I'm constantly moving this sodium into the peritubular fluid, so the fluid between the cell and the peritubular capillary, that means that the concentration of the peritubular fluid is going to be consistently very high in sodium. So every pump, we get 3 of them moving into here, and we have lots of pumps. So there's just oodles of sodium in this space. And another thing to keep in mind is the peritubular capillary, the blood in it is in motion. So it's not just the same fluid sitting in here, which would allow this to equilibrate. We have super high sodium in between the capillary and the cell. And the blood that's moving through is going to be lower in sodium on average, but it's also always moving along. So what that ends up meaning is that this steep gradient between the peritubular fluid and the capillary is going to keep sodium moving into our peritubular capillary. But the system never reaches equilibrium, which would stop diffusion, because as soon as we get the sodium in here, it gets whisked away to somewhere else. And then we have new sodium poor blood coming in to follow. So the movement of the blood in the peritubular capillary is also important to consider because that's going to keep the system down here from reaching equilibrium. And it's also going to keep the interface between the cell and the peritubular fluid from reaching equilibrium. So between this pumping and the movement of the blood, we have a very efficient system set up where we're going to keep the concentration of sodium inside of the PCT cell low due to pumping. And that creates a situation where there's consistently higher sodium in the interstitial fluid than there is in the peritubular capillary. And once that diffusion takes place due to that steep concentration gradient, the sodium is immediately moved away by the bulk flow of blood to make way for new blood, which is low in sodium and that keeps the diffusion going. So this system that we set up with this pump is efficiently self-reinforcing, which is important because we want to use these sodium gradients to do work. So if I know that we're putting sodium back in our blood, and probably there's other sodium in the blood as well. So maybe you just ate a whole thing of McDonald's French fries and your blood sodium is like through the roof. That's going to come out in your filtrate, which means the tubular fluid up here which in retrospect, I probably could have left myself more room for, but whatever. So the tubular fluid is going to be consistently higher in sodium than the inside of this cell. So another gradient set up down here that's transferring to up here, the apical surface that we can use to do work. And work is what we want to do. So some of this sodium transport is going to be passive just because of how steep the gradient is and how small sodium ions are. So via sodium channels, we're just going to have some amount of regular old diffusion. So I'm going to make myself a little key over here in this corner. So any dotted line is going to be diffusion, which we of course know is passive. And then we're also going to need a solid line, which is going to represent reabsorption, which it's not passive. And then we're going to use another color for secretion, also solid. So those are only the reabsorption and secretion are only referring to the direction of movement and not to the nature of the transport. So what we're going to do is use orange for counter transport, which I've abbreviated here because I ran out of space. And then we're going to use this nice magenta color for co-transport. So counter means opposite direction, co means in the same direction. Okay. So now that we've got that all set up, and we also know that we have this pretty steep sodium diffusion gradient also set up, we can use that sodium gradient to do work, which means we can use it to transport other stuff that we either want to dump or get back. So let's do some co-transport first. So we're going to have in the tubular fluid, this little hexagon, which represents glucose, which is a hexagonal sugar molecule, a monosaccharide. And this is important to put back in our blood because it's fuel that we can use to generate ATP. And so peeing it out is just basically throwing away money. Don't want to do that. So we're going to co-transport it with sodium, which is going to really effectively use that sodium gradient that we set up to move something else. Now, why do we need to co-transport this? Well, number one, glucose is a big molecule compared with sodium, and it's also very polar. It's got a lot of charge and partial charge hanging off of it. So things that are polar, especially strongly polar, and also large, are going to not be able to cross a phospholipid bilayer by themselves. Their polarity makes it hard for them to go through the middle hydrophobic layer of the phospholipid bilayer. And their size just makes physically getting across difficult. So for that reason, they're going to need help. That help can come in the form of active transport, where we're going to pump, or it can come in the form of co-transport, where we don't spend an ATP at the location where we're transporting, but an ATP has been spent elsewhere to set up a gradient. So down here is where that elsewhere is, but we're going to use that energy, that stored energy, in the form of an electrochemical gradient. We're going to use the energy up here. So this is not diffusion. Oops, it is co-transport. And we're going to make the arrow be blue because we're doing reabsorption. So we're reabsorption, oops, accidentally was still on eraser mode. Go back, there we go. So we're going to do reabsorption of glucose and of sodium together using that sodium gradient. And this same method of co-transport is also the method that we reabsorb other nutrients as well. So I'm using glucose as the sort of the flagship example, but what I'm going to say is that, like, let's say it wasn't glucose, it was an amino acid, same process. So we're going to co-transport it with sodium because we set up that sodium gradient. So glucose and other organic solutes, which I'm going to say are primarily monomers, i.e. monosaccharides like glucose or an individual amino acid. So one subunit part of a polymer that is used for fuel or stuff. So the macromolecules that we eat broken down into their little itty bitty parts. So if we're constantly reabsorbing these things into the PCT cell from the tubular fluid, we're also going to create a scenario, because another thing to note is that the apical layer are joined by tight junctions. So probably haven't thought about cell junctions in a while, but you need to. So in this case, the reason that we're not having a situation where transport could also sneak through, so maybe some water or some other solutes could sneak between the cells and get into the peritubular fluid, is because tight junctions are waterproof and solute proof, and they effectively maintain a barrier so that the constituents of the tubular fluid are only going to be able to go anywhere if they go through this cell. They can't sneak around. And that also functions to keep the peritubular fluid separate and discrete, which means that its concentration of things will assist in the movement. So if I'm constantly co-transporting sodium with glucose and other organic solutes into the cell, that means the concentration of those things, the glucose and organic solutes, rather, is going to be consistently higher than in the peritubular fluid, because ordinarily those organic solutes and stuff wouldn't be in there. There'd be no reason for them to be. So that's going to create a situation where once I've co-transported glucose and any other organic solute, as long as I have a hole for it to go through, so in this case, I'm going to make myself a little glucose hole. The rest of the process is reabsorptive, but not co-transport. and is more passive because I've set up this gradient. So as long as I give it a space to go through of some kind, it's just going to diffuse back into the blood. So that's super handy. Now let's talk about cancer transport, because that's another one. And in this case, we are secreting. And I said that for counter transport, we use orange. So let's do that. So this is the same principle where we're using the gradient we set up at the other end of the cell to do work at the apical surface. So in this case, we are going to be reabsorbing sodium again. But we're going to be secreting in the other direction. So the way this transporter works is that the binding sites oppose each other for these substrates. And so the open position for one side, say open to the outside, is not going to have the same components as the open state to the inside. And then we can trade things instead of co-transport them, which is good. So I'm going to make this arrow a little bit thicker, and then we're going to use the red one to say we are secreting whatever this is. And the thing that it is a proton. So remember, protons are units of acidity. So we've talked about the blood buffer system and the role of respiration in managing that. So we use a proton to kick oxygen off of hemoglobin to drop off oxygen. And we also use the bicarbonate to buffer our plasma. We also saw the role of carbonic anhydrase and its reactions in the parietal cells of the stomach to make hydrochloric acid. And we've also got the role of those same reactions and dealing with protons. So protons are consistently higher in the blood than they are in the peritubular fluid. And they're going to just pass through a little leak channel. So they go out of the blood and into our little cell. And then we have some other cool stuff that goes on. And unfortunately, I didn't leave myself quite enough room to draw this, but I will explain it. So if we know that the filtrate comes from the blood, we can assume that blood buffering stuff happens in the filtrate as well. So we're going to have basically conversion of bicarbonate and a proton. They'll associate into carbonic acid, and that'll then get pulled apart into CO2 and water in the tubular fluid. So we have the end result, CO2 and water, which I'll draw up here. So the CO2, since it's consistently going to be higher in the tubular fluid than inside of the PCT cell, it's just going to diffuse on in. No problem. It's a tiny gas, doesn't have any trouble getting across the membrane on its own. The water, because it's water and it has no problem diffusing across cell membranes and what have you, it's small, it's relatively small anyway, and it's small and angled enough to get through. And also it's only partially charged, not a full ion, meaning that its charge issue is not big enough to prevent its movement. So we're going to have osmotic water flow, because if we're drawing sodium into the cell and other solutes, that's a constant influx of things that contribute to the cell's osmolarity. And if we know that water just wants to be where solutes are, then it would make sense for water to be just following the solutes. So it's like, hey, wait up. I'm also a solute. I just want to hang out with you, so I'm going to follow you. So what this ends up doing is we've got plenty of water constantly coming in to combine with CO2, and that forms carbonic acid, which then turns into its component parts. So carbonic anhydrase reaction, again, in a different cell for a different purpose. So we know that this guy is going to dissociate into bicarbonate and a proton. So in some cases, the proton can come in from the blood and combine with any ambient bicarbonate that's forming. In other cases, the proton can arise from the incoming CO2 and water combining to form carbonic acid and then dissociating. So there's two possible options for where this proton comes from. It can either come directly from there, or the incoming one from the blood can combine with bicarbonate to form carbonic acid, which then dissociates again. So there's the possible routes. But regardless, the result is that we are going to put protons into the tubular fluid from the blood, and we're going to keep them moving into the tubular fluid. So out of the peritubular fluid, out of the blood, and out of the PCT cell into the tubular fluid. And this process also occurs in the DCT, which we'll look at later. But the net result is that we effectively scrub lots of protons out of our blood via our urine, which means that the urine's pH is consistently more acidic than body fluids. And that's an additional way, and also the most powerful way that we manage our body fluid pH is by just getting rid of excess protons in our urine. So again, with redundant systems, we've got a bunch of different buffering systems that are all working together to keep our blood pH right around 7.4, which is a little bit north of neutral, and to make various basic or acidic secretions at various parts in our body. So parietal cells use this reaction to make protons to combine with chloride to make hydrochloric acid. In the ductal cells of the pancreas and the Brunner's glands of the duodenum, we have this reaction being used to generate bicarbonate, which creates alkaline substances as well. So this repeated reaction that happens all over the body is used in a lot of different capacities to do a lot of different kinds of work, but it all centers around pH management. So here's another place where it does that thing. Okay, the last thing we need to worry about here is our bicarbonate, because bicarbonate is really the most useful when it's in our blood plasma, helping to buffer our blood against potential pH changes. And also that means that this bicarbonate is going to be available for things like the reverse chloride shift, so it can come back into a red blood cell at the alveolus to recombine with a proton and then turn into carbonic acid and subsequently CO2 to breathe out. Or we can use this bicarbonate in one of the alkaline secretions in the digestive system. There's all kinds of places we could use this bicarbonate, but in order to use it and transport it, we have to get it into the blood. It can't chill in this cell. So we have to transport it. That transport is again going to happen due to a sodium gradient. So again, we're going to do some co-transport. So if I know that the concentration of sodium inside of the PCT cell is consistently low, that means that it's going to want to diffuse. And one of the ways that it does that is, of course, due to our pumping. But here's another thing. If we're constantly forming bicarbonate in here and separating it from its conjugate acid and the other two compounds that make it, if I have sodium that's already low inside of the PCT cell, then this motion is due to pumping, right? So the gradient down here at the basal surface is not going to be the same as the one at the apical surface. At the apical surface, I have sodium wanting to move in because inside of cell low, tubular fluid high. At the basal surface, sodium is not going to necessarily want to move out because it's already low inside the PCT cell, and therefore it's going to be consistently high in the paratubular fluid. So that gives us a question mark, like, why does it want to move out here? Well, it's going to be co-transported, but it's going to be co-transported using the gradient of something else. So ordinarily it's the sodium gradient that is sort of carrying this whole shebang, but in this case, we're constantly forming bicarbonate. And so bicarbonate is going to be high inside of the cell and low in the paratubular fluid, which means it just wants to go ahead and be reabsorbed just due to its concentration gradient. And end up in here. And then sodium is going to come along for the ride. Co-transported, capitalizing on bicarbonate's gradient, not sodium's own gradient. And then it'll go ahead and diffuse into here as well. So this co-transport, I'm going to actually take the time to explicitly write. I ran out of room. Let's try that again. Bicarbonate's gradient. There we go. Okay, so lots going on there, as you can see. If you are not feeling super comfortable with this information quite yet, that's a normal experience. Repetition is our friend in, learning this kind of stuff. Oops. And it's probably going to take a few times before you feel comfortable and have enough facility with this to do something like try to explain it on your own without looking at resources. So all that is just to say, if your head is spinning right now and you're like, man, I can't learn this, you can, absolutely. It's going to take some repetition. So you're not doing it wrong. It's just going to take some elbow grease. That's kind of the deal with this particular chunk of material. So what I would say is keep touching base with this material, read it again, look at the picture again, watch the video again, bring it back to Zoom and ask me if you're one of my students. And every time you do that, one more piece will kind of fall into place until you have a full picture of it that you can reliably communicate by yourself. And that is where I'm going to stop recording. So hopefully you found that useful. And